In this video we will discuss about this IMPDH2 localization in the nucleus cause PARP1 apoptotic cleavage. In the previous video we have understand that IMPDH2 localization in the nucleus prevents the cells from energy deprivation stage, and in this results or in these experimental figures the researchers has presented some more insight about IMPDH2 localization in the nucleus, and they have find out is that IMPDH2 localizations will cause PARP1 apoptotic cleavage. So how they come out to this conclusion, we will understand those results in this video. So in the panel A, what they did is that they treat the cells with adipocytes that will cause DNA damage, and then again inject this wild type IMPDH2 in the knockout cells, and in other case they inject the modified IMPDH2 which has this NLS sequence. And what they are able to find out is that cleaved apoptotic PARP1 signals will rise in the cells which are treated with adipocytes, and these results are done in the cells which are treated with adipocytes and then move into the adipocyte free media for 24 hours, and they are able to see these apoptotic cleaved PARP1 signals in wild type cells, in the knockout cells that are supplemented with the wild type IMPDH2, the knockout cells that are supplemented with this modified IMPDH2 that has this NLS sequence. But one more thing that they are able to see, that the knockout cells without this adipocyte treatment will also cause this apoptotic cleaved PARP1 signals. So this made them to analyze the reason behind this, and in the next step what they did is they treat the cells for the short duration of time with high concentration of adipocytes, that is for 2 hours they treated the cells with 5 micromolar adipocytes, and then leave them in the adipocytes free media for 24 hours with 3 different conditions. First, without any supplementations of IMPDH2, and next with the supplementation of wild type IMPDH2 in the knockout cells, and the supplementation of modified IMPDH2 which has NLS sequence in the knockout cells. And still they are able to see the cleaved apoptotic PARP1 signal in the cells that are cells that are supplemented with NLS, both in the case of adipocyte treated conditions and without adipocytes treated conditions. Whereas in other case, such as wild type and the knockout is supplemented with wild type, the signals are very low or negligible, indicates that DNA damage repair done in those cells, whereas the DNA damage repairs is not been completed in these cells, as the apoptotic PARP1 cleavage continues to go on. And after that what they did this immunofluorescence staining and quantifications of this PARP1. So in the immunofluorescence staining they are able to see that even without adipocytes they can see the higher signals for this PARP1 compared to the knockouts that are supplemented with the wild type IMPDH2. This indicates that the PARP1 cleavage or PARP1 PARP1 presence in the knockout cells that are treated with the NLS1 is higher, whereas these knockout cells that are treated with wild type IMPDH2 does not have too much PARP1 signals, and these signals are for the cleaved cleaved PARP1. And when they quantified this cytosolic PARP1, they are able to see that knockout cells that are treated with NLS has higher value of the PARP1 even when it is not treated with the these adipocytes. This indicates that the cleavage of this PARP1 continues to occur as soon as IMPDH2 localizes into the nucleus. Thereafter, they check after checking these PRP1 signals, they also wanted to check whether the apoptotic factors are also increased in those conditions. So when they check this cleaved caspase 3, which is the marker for the apoptotic uh, pathway, and they can see that in the knockout analysis sequence there is an increased cleaved caspase 3 even when it is not treated with adipocytes compared to its the knockout cells that are treated with the wild type IMPDH2. So this indicates that apoptosis will continues to rise as soon as IMPDH2 enters or localizes into the nucleus. And they checked quantified this cleaved caspase 3 in the panel F, where we can see that the knockout cells that are supplemental with the modified IMPDH2 that has this NLS sequence has high base value of the cleaved caspase 3 compared to the wild type conditions. And so now, right now they made a hypothesis that IMPDH2 will localize when IMPDH2 localizes into the nucleus. It interacts with the NAD plus cells and will cause a decrease in the NAD plus amount and thereby decrease in the polyadiporibose, and thus the energy consumptions by other enzymes goes down, and this prevents the undesired signaling pathways activated in the cells. So if in order to test those hypotheses, they did one more studies. So that is, they first check this polymonoadiporibose amount in the nucleus, and they are able to see that those amount will goes down in the knockout cells that are supplemented with the NLS. And this is because because more IMPDH2 is in the nucleus and it interacts with the NAD plus cells, and less amount this will lead to the less amount of NAD plus cells in the nucleus, and though low NAD plus amount in the nucleus will cause low polymonoadipose in presence in the nucleus. So what they did is they supplemented this NAD plus in the nucleus in these conditions and wanted to see whether it will cause or it will supplement it will help in recovering of this polymonoadipose in the cells. So they supplemented this NAD plus in those conditions, that is NAD plus supplementations occurs at 0 macromolar, 12.5 macromolar and 100 macromolar in the knockout NLS sequence, in the knockout NLS samples, and they are able to recover polymonoadiporibose partially, but in the wild type it is still higher. So to some extent they are able to recover that adiporibose, indicates that NAD plus has a direct relationship between polymonoadiporibose. And thereafter what they did is they did one more study where they check the NAD plus levels within the cells at different time intervals. That is, once they treated the cells with adipocytes for 2 hours. In this case they can see the decrease in the NAD plus cells in the wild type, indicates that the cleavage indicates that this PARP1 is apoptotic cleavage, or cleavage is not occurs completely, whereas in the case of this knockout NLS they can see that the difference is not significant, indicates that the PARP cleavage is goes on even before this adipocytes gets even before this DNA damage is triggered by adipocytes. And after 24 hours they are able to see that the NAD plus amounts is recovered, or the difference between the NAD plus amount in the wild type cells that are treated with adipocytes and that are not treated with adipocytes are almost similar, are not significant. And also for the knockout analysis The difference now is not significant, indicates that DNA damage recovery occurs might have occurred uh, in the this adipocytes, in the cells that are recovered from the adipocytes after 24 hours, even for the wild type cells and the knockout NLS. And these things are shown in the immunofluorescence image on the right hand side, where we can see that knockout wild type cells, the signals for this NAD plus in the without treated with the adipocytes and with treated with the adipocytes. And for the knockout NLS, the signals for the without treated with the adipocyte and with treated with the adipocytes. So in this case, after 2 hours, this seems comparable, whereas in this case we can see that the intensity is DMSO is higher compared to the adipocytes, but after 24 hours the intensity looks similar for the knockout cells that are supplemented with the wild type IMPDH2 and the knockout cells that are supplemented with the modified IMPDH2 that has this NLS sequence. So this indicates that their previous hypothesis also, that localizations of the IMPDH2 in the nucleus will cause lower amount of NAD plus, is been validated again. So the final outcome, that they conclude that IMPDH2 when localizes into the nucleus will cause PARP PARP1 apoptotic cleavage, irrespective of whether there is a DNA damage or not, because you can see that in the case of knockout NLS, the PARP1 apoptotic cleavage is for signals are still visible whenever, even though they are not treated with the adipocytes. So I hope you understood their final conclusions, that role of IMPDH2 towards this PARP1 apoptotic cleavage, and thereby helps in the maintaining the cells, in helps or prevent the cells from moving into the energy deprivation state. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.